Well, good morning, everyone. I'm delighted to welcome everyone to our soil science seminar. I'm especially delighted to introduce our speaker, Yunjiao. Zhao is from Nanjing, China, so the southern capital of China. She earned her BS degree in 2018 from Hohai University in Nanjing, a very well-known university. She came here, earned her master's in 2021 in soil science, and now she's going to talk to us about her PhD. Ms. Zhu.
Thank you so much. Today, my topic is impact of drainage on soil nitrous oxide surface flux and subsoil concentration. We will introduce some insights from a multi-year field experiment and a lab-controlled experiment. A little bit self-introduction. I am a soil science PhD here. I got my Bachelor of Engineering in Agricultural Water Conservation Engineering in 2018. And after that, I come here. So this is my 6-7 year here. I got my Master of Science in Soil Science in 2021. And now I have my committee member, have Dr. Castellano, Dr. Sotirios Akontolis, Dr. Robert Horton, Dr. Brian Emmett, and Dr. Philip Dixon. So a little bit background about drainage first. So 150 years ago, around 1850s, we all know that Iowa was a prairie. It is like a wetland. The left photo here shows like at that time, if you want to pay tax, you need to hunt, and a lot of people hunt with their boat. And the right photo here is a piece of very special tile drainage dated in 1885. It says, we the men who started the tile work did so with the motive to benefit the town and the country. So since 1880s, around 150 years ago, the Iowa started to have drainage system. And that is one of the very important reasons why Iowa 150 years ago was a prairie to today's very important agricultural state. However, our drainage system is failing. This is what my boss, Dr. Castellano, will tell me. And we need to redesign and re-management the tile drainage. And in this case, we have a lot of things to concern about. One of things nowadays is nitrous oxide. This is actually a little bit of drawing I drew for a lot of like 10 years of kids for adventure of nitrous oxide. I say little nitrous oxide is trapped in the underground soil. Would you please help him get out of there? So you can see in these photos above the soil, we have plants, we have flowers, we have butterflies, we have fungus, we have a lot of things. But when I draw this, I was thinking, how can I draw below the soil? What can I do there? And I was trying really hard, so I got a little bit of earthworm, a little bit of root. And anything else, I don't quite sure. So this thing I want to say is, yeah, I know it is for kids, but I want to get a little bit say, we don't know a lot below the soil. We have no idea a lot of things, what's going on there. This actually inspired my question here. My question have two parts. First, surface flux. On the surface of the soil, how do drainage systems affect the soil nitrous oxide emissions? And for the subsoil, what's going on there? To answer these questions, we have two parts of experiments. The first is a field experiment. We have a huge field called drain space. In this drain space field, we use 5 treatments here. We have known drain, we have conventional depths, and median spacing draining. So when we talk about the depth and the spacing, the depth is the depth of the tile drainage, and the spacing is the space between the two tile drainage. And in the conventional depth and the medium spacing drainage, the depth is 1.2 meter, and the spacing is around 18 meter, and the intensity for it is around 1.3 centimeters per day. For conventional depth and wide spacing, the depth is still 1.2 meter, and the spacing is around 24 meters, and the intensity is around 2.5 centimeters per day. And then we have shallow depths and medium spacing. The depth in this case is only 0.8 meter, and the spacing is medium, so it is 18 meters, and the intensity here is 1.3 centimeters per day. And we also have shallow depths and narrow spacing. Shallow depth is 0.8 meters, and the narrow spacing is around 12 meters. So it gives me around 2.5 centimeters per day, the drainage intensity. So in this case, in the field, we have surface nitrous oxide flux measurement. From April 2021, we start collecting soil surface nitrous oxide flux in 4 drainage systems: no drain, conventional mediums, conventional wide, and shallow narrow. And we have 1 fertilizer rate in this case, 230 kilograms nitrogen per hectare. We choose this because it is like a normal range for Iowa farmers, and we use this little machine. This is a Lycor smart chamber. We start from 2022. We use smart chambers and nitrous oxide analyzer to give us readings. One of the things is it can read every second. So we don't need to cover the soil top for a long time. We just need to cover the top for only 2 minutes. It really helps us to not change the upper boundary of the soil surface. We measured year-round in 2 treatments, in no drain and conventional medium. So in these 2 treatments, we have data from all years. And we have subsurface nitrous oxide concentration measurements, in which case I think it is one of the most interesting in this experiment. So from December 2022, we start collecting soil gas samples to measure nitrous oxide concentrations at 5 depths: 5 centimeters, 15 centimeter, 30 centimeter, 60 centimeter, and 90 centimeter. So we pound this kind of little tips in the different kind of depths, and we use a syringe to get out of the tube. So what is the air? Traps this. So this is actually a tip. I cycle them from the previous experiment. So it's probably a little bit dirty, but you can see how it looks like. Also, we have soil moisture, soil temperature, soil water potentials, and we do have soil moisture data at different depths using PR2 sensors, 10 centimeter, 20, 30, 40, 60, and 100. Okay, results. We are still in the data collecting phase, so I will show some of the results here. So surface nitrous oxide flux measurement since April 2021. This is a time series data since 2021 to by the end of 2023. So in this case, we can see the little arrow here, they are fertilizer time. So after the fertilizer, the nitrous oxide flux will go up very, very quick. After 2 weeks of the fertilizer is the most important time in the whole year. And one of the things is you may think is a little bit different is 2021. We don't really have a big peak. Why? Because 2021, we actually use GC. So we need to cover the top of the soil for 45 minutes. Every 15 minutes, we take a data. So it is not as accurate as the later years because we can have measurements every second and only cover the soil top for 2 minutes. So in my thinking is the traditional method, the chambers and the GC really underestimate of the nitrous oxide flux in almost a lot of research. And also a little bit I want to say is in the little red circle. In this little red circle, it is after the spring thaw. They have a little bit of spike here. Compared to the fertilizer, it is nothing, but it still have an increase there. We're trying to catch them like twice a week and make this measurement. And which something is not in here, but I would like to mention is this year, I don't think we will have anything like this. In the past whole winter, it is very warm. We got nothing. So this year is very different. So for the cumulative nitrous oxide emissions over the past 2.5 years in the 2 treatments, so we have conventional medium in the black one, and we have the known drain in the blue one. So you can see the conventional medium has way less nitrous oxide emissions compared to the known drain. So conventional medium, averagely each year is like 8 kilograms nitrous oxide nitrogen per hectare. However, the conventional medium is around like 14. So it is a huge difference there. And a lot of them, it just come here in around 2 weeks. So that 2 weeks is the most important time in the whole year. It is crazy. And we can easily see here too, this is a cumulative nitrous oxide emissions over the past 3 growing seasons in 4 treatment. So in this case, we just compare the 4 growing seasons, several growing seasons have 4 treatments. We have CM, which is the conventional depth and the medium drainage. And we have CW, conventional depth and wide spacing. And we have SN and shallow depths and narrow spacing. Of course, we have known drain. So in the other 3 treatments, we did not detect a very big statistical difference in the 3 other drainage systems, but the known drain is almost like double of the other 3 drainage treatments. Why the other 3 does not have difference? One of the things I was thinking is from 2021 to 2023, we all know that it was very dry. We barely saw any drainage in the tile pumps, the sumps. We did not see anything except the first several weeks of May. So I think that's probably one of the reasons we cannot detect a lot of difference in the other 3 treatments. Also, this is a figure kind of easier to show the cumulative nitrous oxide over the past growing season. It's like a comparison. So the purple one is 2023, the green one is 2022, and the orange one is 2021. In the known drain, in 2022 and 2023, they all have around like 20 kilograms nitrous oxide nitrogen per hectare. It is a very high number, but I would not say it is impossible. First is around the literature. I don't think there is a lot of treatment really do known drain. And in our case, we put curtains between the known drain and the other drain, so there is no water can go out most of the case. It is a really known drain field. Also, I do think the machine have a big difference too. So in general, all the results coming from the nitrous oxide emissions is no drain really have a big difference. So in this figure, it is a subsurface soil nitrous oxide concentration at 5 depths since December 2021. December 2021, the winter of 2021 and the early winter of 2023, we have some very cold days and then thaw, some cold days and thaw. So in this case, you can see in winter, they actually have way more subsurface soil concentration than summer. The surface is almost close to 0, but in the winter time, it is very high. And also the other thing I would like to mention is CM, the conventional medium drainage. You can see the highest is the red line, and the red line is 15 centimeters. And the known drain here, the highest concentration happens in this yellow line, which is 30 centimeters. So in this figure, we are better to show the time series analysis by depths. We have 5 centimeter, 15 centimeter, 30 centimeter, 60 centimeter, and 90 centimeter. So we see like below the 90 centimeter, most of the thing happens between the 5 centimeter to the 60 centimeter. So in 2024, we just install those kind of tips from 5 centimeter to 60 centimeter, see what's going on more in this area. And also in the winter time, it's still higher. In general, it's higher than the summertime. And also in the 15 centimeter, the CM, the red line, have more concentration than the known drain. However, in the 30 centimeter, the known drain have way more concentration, it's way higher than the CM, the conventional medium drainage. So from the subsurface gradient, different concentration, different depths, and from the top of the flux, we are trying to do some analyze. But the first step is we are trying to estimate the nitrous oxide emissions diffusivities. So in this one, it's a very, very first step. We just assume it is steady. It is not the same, it's not correct case, but it is a good one to do the first step. So the diffusivity from the 15 centimeters to 30 centimeters, and the diffusivity from the 5 to 15 centimeters. We can see in the winter, it's way lower than in the summertime. And in the summertime, in the summertime, which is after this, in this case, the CM most of the time have like a higher diffusivities in both depths than no drain. But we're still trying to figure out how to calculate. We plan to calculate in a transient gas flow and calculate the diffusivity then. So in our very preliminary conclusion so far is first, the non-drain soil has a significantly higher nitrous oxide emissions than drain fields, and the drainage reduced the nitrous oxide emissions by around like 8 kilograms per hectare per year. So this is like a little figure, if you want to remember anything, is the non-drain have higher, way higher. And the second thing is, most nitrous oxide emissions were produced around 0 to 60 centimeters of soil, and non-drain soil had higher subsoil nitrous oxide concentration than the conventional drain soil. However, the non-drain soil concentration peaked around 30 centimeters, but the CM, the conventional medium drainage soil, peaked around 15 centimeters. And during the winter, the soil nitrous oxide concentrations are large, but the surface flux are small, which indicates low soil gas diffusivity. It maybe come from like low temperature, decreased airfield porosity, and something we need to think about in the future. So that is like our field experiment side, and we do have a corresponding column experiment. This is a photo of what the experiment looked like yesterday. It is happening now. So this is in the inlet USDA rhizotron. So we have 12 this huge soil columns, and we try to control the things. What's going on, the temperature, the water, and a lot of things, what's going on here, and we try to understand how does the drainage affect nitrous oxide in this case. So we collect soil from the field next to the drainage, the field experiment field. We have Okaboji, we have Clerium, and we have Nicolette. So we have total 12 columns, which including 3 soil type, and we have 2 treatments. So in this case, we use the treatment as like a flood time lens. We flood the soil columns from the bottom to the surface, and then half of the column stay there, only saturate them for overnight, and half of the column saturate for more than 3 days, 3.5 days actually. And we drain them, and we drain from the bottom, and we see how much the soil nitrous oxide come from the bottom, from the surface, and what is the subsurface soil nitrous oxide concentration at different depths. This is our design. So the soil column, each of them is around 25 centimeters in diameter, and the length is 60 centimeters. The 60 centimeter one is because from our field experiment, we saw the most important critical zone is from 0 to 60 centimeters. So in this case, we have gas port. It is different from the gas port we get in the field. One of the things is it is a Gore-Tex probe. So I want to specifically mention this because in the field, we really measure the gas here. But in this case, Gore-Tex only allows the gas come in, but the water cannot come in. It causes something like a membrane. It can Actually, and we know that nitrous oxide can easily dissolve from the water. So we are measuring different things. If you want to see this, we install them in different depths, use this kind of material, try to get the gas port concentration, and we also have TDRs, measure the temperature, measure the soil water content, and we install the tensiometer too, try to see the soil water potential difference at the different depths. And we also vary a lot of nicemeters in different depths too. So we are trying to get the soil water to see how does the nitrate difference in different depths. So in this little bottle, we can see, so this is a bottle we're trying to flood the whole soil columns, and we have a lot of water in the bottle, and from the bottom, we get a lot of water submerged from the bottom and lift up to the surface. So we're trying to create an artificial flood or rain or non-drain occasion to see what's going on. And we try to measure the gas ports. So in the surface, we have this kind of auto chambers. The auto chambers can give us nitrous oxide readings every 1 hour. And we also get carbon dioxide readings every 1 hour. And for the gas port, we have to do manually, get some injections, get some measuring. We're trying to do that around like normal time. We do that every day, but if we drain them that day, we will do one measurement before the drainage and one measurement 6 hours after the drainage. So this experiment is actually still going on. This is like day 15 of the experiment today. So I will show a little bit preliminary results here. It's kind of hard to see. So we have 3 kinds of soil. We have clarion, we have nicolette, and we have okoboji. 3 kinds of soil with 3 kinds of color. The red one is clarion, the green one is nicolette, and the blue one is okoboji. We measure the soil gas concentration over the time from 0 to day 10. This is the first round of the experiment, so it's only happens after it only ends after day 10. So in the upper here, it is drain them very quick. We drain them after flooding half day, overnight. But here, it is no drain. It is like we keep the flood situation for more than 3.5 days. And the dashed line here, it is a drainage time. So before the dashed line is all flooded, after the dashed line is all already drained, and the drainage keep going. So one thing is, I would say, very easy to see is soil type. It's very different. So in the clarion, so in general, it does not have that much a peak. But in nicolette, so the y-axis in the non-drain here is actually over 10,000. It is very huge. It is a big difference compared to the drain and the non-drain. And the soil type here is a big difference too. So which means the clarion actually does not have a lot of emissions and the concentration and the productions in that case. But the nicolette have huge. And the other thing I would like to mention is in the drainage case, so we only flood it for overnight and then drain very quick. You can see the concentration is actually getting increased in different depths. So the first one is 10 centimeters, second is 20 centimeters, and here is around 50 centimeters. It's getting increased over the time after the drainage. And for the non-drain here, it also has a similar trend, but it will like decrease soon, a little bit more quicker than the drainage field. So this is just a preliminary data. We have a lot of things going on, and a lot of things I would like to say, but it is still very not that mature. But I want to share with you because I come out yesterday and I was very excited for this. And for some future work, So this is actually a picture from like 100 years ago, and they bury the tile drainage, dig very hard to bury them to install the drainage system in Iowa. And now I would like to say I would like to dig more deeper inside of the soil to see what's going on there. Thank you so much. And if you want to talk with me, this is our email. You probably know everyone here, but please contact me if you have any questions and suggestions. Thank you.
Thank you. Questions for Ms. Zhu?
Yep.
Really, really good. I very much appreciate what you're doing. I have enough questions to last most of the morning, but okay, you said, okay, so in the field study,
Yes.
it was relatively dry across the 3 years.
Yes.
You also, you measured water potential and water content.
Yes, we did.
Okay, so what was the difference in water content or potential between and drainage?
It's actually still have a lot of difference, especially in the first like around like 20 to 40 depths. They have the most difference there. And I would say first is like the surface is dry, that is true. But after when it's getting to like 20 centimeters, we can see the big difference between the two fields. I did not quite share here, but I do have the data. Yeah, so, but like for the deeper, like 100 centimeter, the difference will getting smaller too. Yes.
So I really appreciate that you're looking at different soil, recognizing differences in soil. But what, for your experiment, what's the difference between the Clarion, Nicolette, and the Koji?
It's a like I remember it's one is like a clay loam, one is like a loam, one is like surface is a clay loam and the clay and the other depth is like the clay loam. The main difference I would say is the soil texture. As far as we see, the soil texture have a big difference there. I think like the textual analysis, well, it's a like the particle distribution is a very important thing for the gas movement. So like for example, in the Nicolette, in some of the columns here, in actually that one in the number 6. It's a Nicolette and number 3. It's number 3 and number 6. Both of them are Nicolette soil. It's very, very, very slowly flood. So I do think the soil type first affects the gas movement, affects the water movement, so affects the nitrous oxide production in everywhere.
Okay, so you're focusing on the particle size and distribution of pore spaces then?
Yes, that's what we feel. Like we were trying to get more like biologically, but I feel like the whole experiment is more like a soil physics way. So we don't have a lot like microbiology, like what is a microorganism there difference. So we did not focus on the other. I know they have a big difference there, but we did not focus on that part.
Just final follow-up, what's do you think might be the relationship between the soil in these columns and the soil in the field? What I mean is that the field, there's probably some structure to the soil that
Yes.
provides pore spaces.
Yes.
Did you do anything to try to preserve those in making these columns?
Yes, when we make this kind of columns, we use hydraulic probes, and they basically dig everything right, look like that in the field, and try to avoid any compaction. Unfortunately, probably one or two columns still have a little bit compaction on the top, but we try to preserve as many as I can as we see in the field. It was not a very easy work.
Sure.
Yeah. Yes.
I'm going to ask a question from Dr. Horton.
Yep.
Dr. Horton asked, in the field study, do you know the depth of the water table in your treatments? Part one, water table. And number 2 is, and do you know about ice content in the soil?
Thank you, Dr. Horton. So I would say first, the soil water table, we do not have a direct measurement for that. we have like some estimation because we have the water management, we have the soil moisture content. So we say like if it is like already like very stable for like 1 week or something like that, we say, oh, it's already saturated that. But yes, we do not have a direct measurement for water table. And for ice, yeah, thank you, Dr. Horton. That is a very good point. It is, yes, we do not have a good measurement for ice. So that's one of the reasons I do not quite share the moisture data here, because I'm still thinking about how to deal with that. Because in the winter, a lot of time we have ice, and what we detect is only liquid water. And how does that affect me? And how does that affect like some, like here? Like in here in winter, there must be a lot of ice there, and I just have the liquid water content. How can I use that to explain a little bit in the winter here? It's a little bit tricky for me. So, thanks.
Then I have a question about slide 11. Yeah, so,
Yes.
So this was really neat when you were showing this, and you mentioned the different methods. And you mentioned the main peak.
Yes.
And then the circled peaks.
Yes.
But I'm curious in 2022 and 2023,
yes,
why there are two secondary peaks each year after the main peak.
It's after the rain. It's
okay, each rain event.
Yes. So we try to, after the rain, next day we will go to the field and measure that, so we can catch the peak of the rainfall there. So in the early July, we can still detect a lot of like the rain peak, but later it's getting drier and drier. It is not really there. Yeah. Dr. Thompson?
Slide, I think you were saying that you thought GC method was less accurate.
Yes.
Than the method.
Yes.
Could tell us a little bit more why you think it's a bit more accurate. It seems like GC method is integrating over. So that's really comparing apples to oranges from the beginning. It's integrating over a long period.
Yes.
But is there something about the way the instruments are calibrated that's different? How is the instrument you're using calibrated? Just tell us more about that.
Okay. Oh, I'm not like trying to say GC itself. I would say like traditional chamber and the GC together. So the traditional method we cover with the top of like in my case we use a 30 inches big round rectangular chambers. So we cover that for 45 minutes and we take a gas sample every 15 minutes. We did that exactly the same in the 2021. So this can this have, I think first is we cover the top for 45 minutes, which is easily the gradient in the chamber is getting lower and lower. So it will like plateau. So in my experience, like we actually try to use a light cord to measure like 20 minutes or 30 minutes what's going on there, and we actually see it will getting plateau after like 15 20 minutes. So in this case, if we just take three measurements in the 45 three or four measurement in the 45 minutes, it's like it can be like 1 2 3 4, and we have like a regression curve. We think the plateau will actually affect a lot in the measurements, because when we do the regression, a lot of time we don't really know if it is plateau a lot or not. And when we do the regression, I think a lot of time we use the linear. It's very common we use linear. I think it underestimates the plateau area. I think like after the 45 minutes cover the soil top, the gas gradient is already small, and they do not come out a lot. And also is every 15 minutes, and we take like a small amount of the samples, and the chamber is actually huge. This chamber is around like the area is around like 3,000 centimeter square. So, and every time the little vials we use, it was only around 10 ml. We get the 10 ml syringe there. It is a small amount, I would say. And so that's one of the reasons I think it's probably is not that representative of the whole chamber too.
In your field design, where was the no drain compared to the others?
In my field design, are you talking about how do we design them differently?
Yeah, the plot layout.
I want to show you this photo. This like the top is like no drain, the bottom is like no drain. The field itself, it's like similar to the other field, but we do have a curtain between the known drain and the other drainage. So the water cannot go out of the known drain field. The curtain depth is around 1.2 meters too.
I'm a little bit
Yes.
concerned, well, diagram you have with your columns,
Yes.
where you have a bottle
Yes.
with water that supplies water to hydrostatic pressure. The column,
Yes.
would that same principle not work in the field, even though you have that curtain? Will not any difference in hydraulic pressure on one side of the curtain to the other result in water movement?
Yeah, I do think so. I do think they have difference, and especially in this, like this helped me understand a lot of difference there too. Like we try to drain from the bottom, actually the water come out only around like the top 20 centimeters. It does not come out that much. It's actually way higher than we thought. So my thinking is, yes, it's different. It's very different. It's very, very different in a lot of cases. But my thinking is like, what can we learn from this kind of case? What is the main point of this case? What is the main point of a not very realistic field experiment and a not very realistic column study? So I think like we're trying to create like in the field, we try to create like a known drain, and we also want to like focus on the middle of the field. So each field is actually around like 120 feet in the length. So we want to do a very, very in the middle of the point to avoid the edge of that. And for the column study, it's something like we are trying to see the treatment difference, more focus on the treatment difference. I know it is not realistic in the field at all.
I'm not criticizing what you've done. I think the point you made, I think, is very good. You're trying to see.
Yeah.
And how you're thinking in a way that we're going to do this with you.
Yeah.
And address those practically.
Yeah. Thanks.
Rather than because then, how was the field drained around these spots? What was the drainage water table depth was at?
Yeah, like in the known drain, in like after the May, a lot of time it rained. It actually the water table, we saw it is like above the surface. It's like submerged under the water. It's like flooded in a lot of area. And for the other drainage field, we have the drainage like working like buried in this way, and they have like sumps in the other side. It is, and yeah, it is a good question, and I don't have much thinking there. Thanks.
That's I mean, you're setting an experiment in probably a drain field, and it's really, really difficult to separate water effects.
Yeah, it is. It is.
So I'm not being critical of what's done.
Yeah. We know that part. We don't have a lot of good resolution for that, I would say. The first thing I would say is we're trying to focus on like the middle of the field, right middle, right on the water table, and we do have chambers between the water, we have a line here, like the drainage line here. We have chambers on the line, and we have a chambers in between of the line and the border to see if they have a difference there. We do not see a big difference there, and a lot of time we do not quite sure it is because of the spatial distribution. spatial difference is huge in this case, or the water table is something we should detect and we did not. Because we will assume the water table at the drainage tile is the lowest, and the other one is like higher. But in the experiment, we unfortunately we did not quite see a big difference there.
So Dr. Horton has a follow-up question in the chat, but also has his hand up. I think he can hear us. Just so you know, Dr. Horton, we're not quite sure that the speaker may just be on this laptop. We'll give it a try. Go ahead and unmute and we'll see if he can hear you.
Can you hear me okay?
Yes, I can.
I wanted to kind of just step back a little bit from your result. First off, great presentation
Thank
and
you.
very, very interesting
Thank
study,
you.
field and lab. So congratulations to you and your team.
Thank you.
I wanted to step back just a little bit and ask a little bit, more general question about nitrous oxide flux measurements. We used to have a colleague in agronomy some years ago named Fred Blackmer, who did quite a bit of work on nitrous oxide fluxes, and he found a large, spatial variability in his measurements. He said they were like a log normal distribution of the fluxes within a field. And I'm just a little bit curious, like within each one of your plots, how many places did you measure nitrous oxide flux?
Yeah, it is a very good question. First, second is it is huge. So the first thing is I would say it is huge, and sometimes make me feel like it is impossible to do such an experiment in the field. Also in the lab, it is very huge. So in the summer, we have the larger chambers, the rectangular chambers. They have 30 inches in the length, so they can detect from one corn row to the other corn row. So there we have 2 in each plot, and they make total for 32 chambers. And in the winter time, we have 4 round chambers instead of 2 big chambers in 2 treatments. So they make us 32 chambers again in the winter too. And it is still huge. It is huge difference, and a lot of time it's very, I will say it's a challenge to deal with that, because I even talk with my Dr. Castellano and say, I don't know, like there is a field, this non-drain field, this field. It is super wet. The corner on the southwest, the southwest corner, the non-drain field, it's super, super wet. It is like every time they have the highest, it's have water flood that all the time. It's very hard to walk. I don't know how to deal with that. And my boss have 2 suggestions. One is like, first, it is what it is. We have 4 replicates and we should average them and see what's going on. And second one is one thing I am thinking like next step I will do is I don't treat them at treatment difference, but I treat each of them as like a different spots, and different spots itself have like soil moisture difference, soil temperature difference, and how does that soil water affect different spots rather than the treatment? I think that's the next thing I will do. And I will say like even in the lab case, we have big difference too. It's still a very tricky part, and I would like to see the preliminary results I got just yesterday. So in this results, like in my case, I found out like clearing is pretty like several replicates, they look similar, and but the Nicolette and Okoboji, different columns, they can have 10 times difference. It is, and why? Why? Even the water content, because they're flooded. How can they have such a big difference? To be honest, it's very hard, and we are still trying to see what's going on because we have some supporting data like the carbon dioxide. We want to compare like the nitrous oxide to the carbon dioxide. Okay, we haven't saw that yet, but compared to carbon dioxide, we know the carbon dioxide, we know it is just producing, but nitrous oxide is something produced and consumed. So what is the difference there? So yeah, it is. We are trying to understand. Like I would say, first is in general from treatment, and second is from spots to spots, and try to understand them. Yeah.
Good, thank you. I have another question. It has to do with like the temporal variability, and I really appreciate that your what you've called your smart sensor,
Yes.
only has to stay covered for a shorter period of time compared to the traditional way to
Yes.
determine nitrous oxide fluxes. So I think that's a plus, but I am curious how you have to put that smart sensor down yourself, right?
Yes.
It's not automated
Yes.
that you can control when it goes down, but a person has to put it down, right? Yeah,
Yeah.
so you maybe are familiar. Some years ago, my group and Dr. Sauer from Enlay, we used to do some collaboration on carbon dioxide emissions from the soil, and we had these Lycor sensors that were automated so
Yeah.
that they would close whatever we programmed them. So we could get diurnal variations in carbon dioxide fluxes, but I know that there's quite a bit of temporal variability in nitrous oxide fluxes. So, if you only go out there once a week or once every 2 weeks or whatever, there's a lot, you may miss peaks.
Yes.
How often in the field, how often did you go out to measure the nitrous oxide fluxes?
In the summertime after the planting, we try to do that at least twice a week, and for at least 3 months until probably like the middle of the August, something like that. But in the winter, we try to do at least every week. One thing is winter is cold, so sometimes we just cannot go there. Like this January, we have a big snow. We just cannot, and also it's like in the summer we're trying to go there around like 8 a.m. and to the middle of the day and try to detect that window. But in the winter it's kind of cold, so we most of the time go to in the afternoon, like 1 to 4, 1 to 5. So yes, the diurnal pattern is very important. I know we miss that. That is true. That is a thing we do not have. That's true. One of the reason I want to say a little bit for myself is the field does not have electricity, so we cannot have automatic chamber there. Yeah, it is a big question. And for the unfortunately, not or fortunately, in the lab control experiment, we are in a racetron and we control the temperature to make them have almost zero diurnal patterns there. So, and we measure them every 1 hour. So it will be interesting to see if we see any diurnal pattern in the lab, but we haven't gathered the data yet, and that's definitely something we will look at.
Very interesting. Thank you so much.
Thank you. Dr. Thompson?
The lab study, I have two questions about this.
Yes.
First one, in this study, is nitrogen soils before
Yes, we do. And sorry, I forgot to say that. It's 50 ppm nitrate, potassium, nitrogen. So the 50 ppm is from the nitrogen. We chose array because we saw them in the farmers in the field. One thing is we apply them in a solution with calcium sulfate and calcium chloride to make it a little bit like a salt solution, and apply them when we flood them.
dissolve
Yeah.
nitrate in the water that you're flooding the
Yes.
soil with. Oh, I see.
Yeah, so it is very, very non-realistic, but we want to see like how if we apply the sub things for them and how do the nitrous oxide react. So that's one of the reasons we measure the nitrate too. Yeah.
Okay, and then I'm going to follow up on this question. You think that barium, Nicolette, and Okachi might also differ in organic matter amounts, and how would organic matter affect nitrous oxide?
Yes, we do think about this question, and unfortunately, we did not have. There are something issue there, so we did not really quite detect the organic matter there. So one thing is the organic matters. I do think my personally is like how does the organic matter nitrify to nitrate and then affects the nitrous oxide? It definitely have a big difference, and also the organic matter, the particle size is it's lighter, it's smaller. How does that affect? Yeah, I do think so. Unfortunately, we were planning to do that, and yeah, I need to say the the fridge was like broken, and the samples are like uh, it's go wrong at that time. Yeah.
Still sample those columns at the end of the.
Yeah, that's a plan after this. We're still in day 15. We have like 5 days to go. Thank you.
Next question.
In the field, how was the nitrogen applied? Was it banded? Was it surface applied and then incorporated with tillage?
We do conventional tillage, so we tillage in the spring and the fall twice. It is it is a continuous cornfield. I think they broadcast urea every year. I'm not sure this year's plan. I will check them with them about that. So one thing is for the fertilizer, yes, is sorry, I like this. So I want to say it. We do see a lot of hot spots, and it's like when we have like a two round chamber next to each other. Some of them just crazy high, and some of them is not. It is super hard to go back to Dr. Horton's question. It's super hard to have the spatial difference there. It's not just soil, and apply the fertilizer is a big thing there too. Yeah, I would like to take any suggestion how to deal with that.
Real quick,
Yeah.
It seems like you have micro hotspots
Yes.
in the identified. I think those that to me would suggest when you get rid of the building,
Thank you. Thank you so much.
Let's thank our speaker again. Great job.
Thank you. Also, my mom is here. Thank you.